This video is an overview of some playlists that I have when I teach Anatomy and Physiology 2 on the heart. And if you'd like more information, obviously these videos go into these topics in greater detail. I hate to be the one to tell you this, but you don't heart anyone and no one hearts you. The ancient Greeks, among others, had this disagreement. Where was the seat of emotion in the body? Some, such as Hippocrates, thought it was the brain. Others, such as Aristotle, thought it was the heart. Aristotle even had a reasonable explanation. He said, if I saw something that scared me or someone who I was attracted to, I would notice that my heart was beating faster. Logically, the heart would then be the seat of emotion. It makes perfect logical sense, but it isn't true. So the heart is a pump. It is an amazing pump when we look at how much blood is pumped in its lifetime over the course of a day. Obviously, blood is a thick fluid, 5 times thicker than water. We have maybe 60,000 miles of blood vessel in our body. Now, that most of these are microscopic, but nevertheless, there's quite a distance here. The heart can regulate the amount of blood which is pumped, changing, based on our body's needs from rest to exercise. And so it is an amazing pump, but it is a blood pump nonetheless, and is not the seat of emotion. It is wrapped in a bag, as is much of the viscera in our ventral body cavity, because the heart is always moving, the lungs are always moving, the digestive organs are always moving, and as such, just as like your foot in a shoe, if it's rubbing and rubbing and rubbing, it could get blisters and adhesions and sores. We certainly can't afford to have that on these vital organs, which are constantly moving across each other. And so here, whether it be in this cat or that, forgive me, I think it was a monkey, might have been an opossum, I'm sorry, I forget. Here you can see that there is a bag around the heart, a serous membrane. Now, there are many serous membranes that, for example, Anatomy and Physiology 2 students will study. Serous membranes are defined by having 2 layers, an outer parietal layer and an inner visceral layer. The outer parietal layer has collagen fibers for strength, but where the 2 meet, we have smooth, simple squamous epithelial cells, which are secreting a fluid, a serous fluid. So we tend to name it based on the individual bag. So for example, with the heart, we will call this the pericardium, as opposed to with the lungs, we call it the pleura, and in the digestive organs, we call it the peritoneum. So there is an outer parietal layer, that's the bag around the heart. There is the surface of the heart, that is the visceral layer. So the surface of the heart is the visceral layer of the pericardium, just as the surface of the lungs is the visceral layer of the pleura. And then between the 2, we secrete a lubricating pericardial fluid. I'm sorry, that Zoom and the internet don't work as well together. I'm sorry for the slowness. So that between these 2 layers, there is a pericardial cavity, and a lubricating pericardial fluid accumulates between them. And so now when the heart is always moving, it is movement between the smooth visceral pericardium, the smooth parietal pericardium, lubricated with the pericardial fluid between them. The pericardium can be clinically important. In some cases, there are tears and the loss of fluid, in which case you can have abrasion. There can be excess fluid, which accumulates there. And so the pericardium, very important and vital in some clinical analyses as well. Let me do this one. And so just as we look at the external anatomy of the heart, To start off with, most of our analysis will be, once we section it. But let's look at this first. The heart is roughly triangular in shape, and as such, we can use the words we use for triangles, like base. So the flat part, the base, is around the second intercostal space. If we start counting the ribs and where the spaces between the ribs, the intercostal spaces are. And then there is an apex of the heart, the tip of the triangle. Now, this apex is not exactly on the midline. It angles to the left. About 2/3 of the heart is to the left of the midline. There's a gene, very important in this, called sonic hedgehog in embryonic development. Yes, there is an important gene named after a video character. And mutations in this, among other things, can cause the heart to angle to the opposite direction. So in general, once again, we look at it as an upside down triangle with the base around the second intercostal space, the apex around the fifth intercostal space, a couple inches to the left of the midline. There are chambers of the heart. So there are atria. These are near the base, and most of the heart is made of ventricles, which make up most of the space, but certainly compose the apex of the heart. And the great vessels of the heart, which enter and leave the heart, they are near the base. So the vena cava, the aorta, the pulmonary trunk, the pulmonary veins, they enter the base of the heart. Now, even on the outside of the heart, we can notice that there are grooves. So there are grooves between the atria and the ventricles. You could call them an atrioventricular groove. Another term is a coronary sulcus. Because the heart is so important and studied all throughout the world, unfortunately, there'll be a couple of places where we realize there's more than one commonly accepted vernacular term for the same structure. So for example, with the heart valves, just as an example, the bicuspid valve, the left AV valve, and the mitral valve, those aren't three separate valves, they're all the same valves, they're just three different names. Medicine is practiced all over the world, and so it just has come up that there are sometimes multiple accepted terms for it. So here we can see that on the outside there are atrioventricular grooves between the atria and ventricles, and then between the ventricles there is a shallow groove known as the interventricular sulcus. Now this is the anterior surface. We would see the same on the posterior surface, and notice that there are blood vessels here. So there's the atrioventricular grooves. Here's the interventricular sulcus, which we can see on the here. And then one last feature that we can see here is the atria just have these small extensions to give them a little more space. They are called auricles. All right, and so on the outside, you can just see a small space, once again, in the atria known as auricles, which give them a little more room as they accept blood. Just a quick note on those blood vessels in the atrioventricular grooves and the interventricular sulcus. There are coronary arteries and veins, and these are so very important. And at first that's counterintuitive because the chambers of the heart on the inside, which we will see, are full of blood. And so if the muscle of the heart needs blood, one could maybe assume, isn't it the case that the muscle of the heart could just extract oxygen out of the oxygenated blood that's in the middle of, let's say, the left ventricle? And the answer is no. It's just too thick. You can't have that much diffusion. And so therefore the heart needs its own arteries and veins. Just as this muscle, in your arm needs arteries and veins, the heart needs arteries and veins. And so on the exterior view, in the atrioventricular groove and the interventricular sulcus, you see the arteries and veins, which are the surface, are the source of the blood supply for the heart. So when the aorta leaves the heart, the first branches are then coronary arteries that go back to the heart. So the muscle of the heart is not getting oxygen from the blood that's within the heart, but rather from arteries which service the heart. There is gas exchange in capillaries, and then there are veins which then drain the heart. The veins end up fusing to form this a single vein that is wider than usual and has thinner walls than usual. And the term we use for that is a sinus. So the coronary sinus is in the atrioventricular groove on the posterior side. So all of the blue deoxygenated blood that the heart muscle no longer wants goes into coronary veins, which empty into the coronary sinus, and then the coronary sinus will empty into the right atrium. So usually, at least when I teach, I often focus primarily on the superior vena cava and the inferior vena cava, bringing blood into the right atrium. But there's actually a third vein as well. The coronary sinus empties into the right atrium as well. So the right atrium gets deoxygenated blood from three veins: superior vena cava, inferior vena cava, and the coronary sinus. When I talk about heart disease later, this is why then lipid plaques blocking these blood vessels is so critical, because this is the oxygen supply for the heart, not the blood that's in the heart. And so then if coronary arteries are blocked, heart muscle then is denied oxygen, it begins to die in a myocardial infarction. So on the outside of the heart, we can see those coronary blood vessels. Now, once we begin to section the heart, we identify three separate layers. So there is an epicardium. This is a very thin, smooth layer. Here, only one layer of cells thick, simple squamous epithelia. And this is another name for the visceral pericardium. So the visceral pericardium and the epicardium, they are the same layer of tissue. So it's very smooth, and once again, because the epicardium then is rubbing against the parietal pericardium, there is no friction, which would develop sores, blisters, adhesions, and the like. Now, on the inner surface of the chambers, the spaces in the heart, there is an endocardium. Once again, simple squamous epithelia, which we could call endothelia. If we wanted to, we could refer to the epicardium as mesothelia. And nice and smooth is good for a couple of reasons. One, if you're going to pump fluid through a chamber, if it has smooth walls, then there's less friction, you need less pressure. Like if you were bowling on a shag carpet, it would be very difficult. The friction would slow the ball down. You would need to push a lot harder. You want a smooth, flat surface. So here's that smooth, flat endocardium, which then allows blood to move with minimal friction. Also, platelets will tend to break and start clots, and start to clump and degranulate and start clots when they hit uneven surfaces. We don't want unnecessary clots forming. That could be the cause of a heart attack or stroke. And so smooth surfaces, once again, better. That's what we see in the endocardium. In between the two, in this cow heart, then we see the cardiac muscle. This is the layer known as the myocardium. Now, maybe for many students in anatomy and physiology too, this was perhaps where you studied, the different kinds of muscle. Maybe just, a quick, reminder here. So skeletal muscle made of huge cells. All right, when you look at them under the microscope, they're so long you can't see where they begin or end. They have this beautiful banding pattern, these beautiful striations. Because they're so big, they have hundreds of nuclei located on the outside of the cells. They are voluntary. So if you think of a part of your body and you try to move it, if it actually moves, you have skeletal muscle there. That is the only type of muscle which is voluntary. It can heal a little bit if injured, not well, but at least it can heal a little bit. In, say, anatomy and physiology too, most of the times you use the word muscle is when you're talking about this kind of muscle, smooth muscle. Here we don't see striations. There are no striations on the outside of the cell, hence the term smooth. Each individual cell is elongated and tapers at the end. There's only one centrally located nucleus. Smooth muscle is all involuntary. Um, and so we have no voluntary control over this. This is what lines most of the respiratory tract. It lines blood vessels. It lines most of the digestive tract, the urinary reproductive tracts. It makes the iris of the eye. It allows your hairs to stand on end. And so we have a lot of smooth muscle in our bodies. And smooth muscle of the three heals the best. All right, so smooth muscle heals rather well. Obviously, as we talk about the heart, we are now interested in cardiac muscle, which is only found in the heart. So this is the only part of the body where this is found. It is striated like skeletal muscle, but it's smaller. Instead of huge cells with hundreds of nuclei, there's only one centrally located nucleus in each cell. It is involuntary. Now, some people would say, no, I have voluntary control over my heart. I meditate and I slow my heart. That's not what we mean. So if you had voluntary control over your heart, you could be listening to a song and say, oh, I love this song. I'm going to have my heart beat along with this song. No one can do that. Now, obviously, when we put ourselves into different emotional states and make different hormones, we make epinephrine, we don't. This can slow our heart down, it can speed our heart up. But then the same is true for the other case as well. When you're under stress, when you're at rest, the degree of contraction in your skeletal muscle or smooth muscle can change. So hormones can influence muscle contraction, but voluntary control is something that the heart does not have. It does not heal at all. All right, so I do not want to have a heart attack today, because if I were to survive it, I would now just have to live the rest of my life with fewer cardiac muscle cells, because the ones that died would not be replaced. So cardiac muscle cells don't divide. I have, as all of the cardiac muscle cells that I have on the day of my death are in my body right now. I'm not going to get any more. The tip-off with cardiac muscle, if you're looking at it under a microscope, is where cardiac muscle cells meet each other. There are thickenings known as intercalated discs, which you can see there. So only cardiac muscle has those thickenings. And at those thickenings, there are I'm sorry, just give me a second. At these intercalated discs, the cardiac muscle cells, they don't end as, say, cylinders here, but rather they form these projections, which then interlock, making a firmer junction. And if we were to look at the types of junctions which exist here, there are two. There are what are called desmosomes, and desmosomes allow the cytoskeleton of one cell to then be linked to the cytoskeleton of another cell. So these tubes are meant to represent cytoskeletal elements in each cell, but here connecting the tube with the protein cadherin is a junction known as a desmosome. So the cytoskeleton of this cell is actually linked to the cytoskeleton of that cell, which is very important. When heart muscle then contracts, these cells aren't going to rip apart. And so there are these physical desmosomes. But then also there is a second type of junction known as a gap junction, where pores actually exist between the cells. So ions, for example, could actually go from cell number 1 into cell number 2. Now, one of the things that is sometimes said is that the heart is not a syncytium. A syncytium is what we do have these occasionally where you have cells which fuse. So you can have separate cells kind of smushed together and become one. The way that the skeletal muscle cells got to be so big with hundreds of nuclei is that when you were an embryo, hundreds of little cells smushed together and became one big cell. So you could have a syncytium. The heart is not a syncytium, but it almost acts as if it was. So the cardiac muscle cells, they're separate. They're distinct cells, but nevertheless, their cytoskeletons are joined. So they're physically joined. And because of these gap junctions, then you can have them be electrically and chemically joined. So even though they are separate cells, they almost act as if they're, one big cell, allowing the heart to contract as a unit and cells not to tear apart. And so cardiac muscle has these types of discs in its, these types of junctions in where cells meet. And under a microscope, you can see that. And if you're trying to distinguish between the two, that's a distinctive feature of cardiac muscle. Only cardiac muscle has that. Now, for an anatomy and physiology 2 student, there were certainly a lot of discussions from anatomy and physiology 1 on how muscles contract, the whole sliding filament theory, action potentials, etc. And that probably took hours. And you might be wondering how much of that material do I have to repeat here as we talk about this new type of muscle? And the answer is not a lot. And the reason is that much of what you've already learned about, say, skeletal muscle is also true of cardiac muscle. So these muscle cells have sodium-potassium ion exchange pumps that pump sodium out of the cells, potassium inside of the cells, and thus create an electrical difference. The outside of the cell is positive, the inside of the cell is negative. And if you were to cross a threshold voltage, so if the cell becomes stimulated, positive charges enter, and you cross a specific threshold voltage, then all of these gates, which are voltage regulated, they open and positive charge goes rushing in. And we can measure that because now the membrane potential jumps into the positive range. We depolarize. Okay. Now, we could explain that more, but cardiac muscle so far is about the same as skeletal muscle would be. However, in skeletal muscle, then once we depolarize, then instantly the potassium gates open and we repolarize as positive potassium leaves the cell. And so we depolarize, we repolarize instantly. And thus this sequence of events and the contraction which results from it, they don't take long. Fast twitch skeletal muscle, this occurs in 1,000th of a second. So this can occur very, very quickly in cardiac muscle in skeletal muscles, in muscle twitches. We depolarize, we repolarize very quickly. Cardiac muscle is different because we depolarize just as we would, but then the potassium gates don't open right away. There's a delay. And then there is a new gate, a calcium gate, which then opens and positive calcium enters, which means that this period of depolarization where we're in the positive round, we hold it. So we hold it, we hold it, we hold it, we hold it. The potassium gates are delayed in opening, and there's a calcium influx caused by calcium channels that we don't see in skeletal muscle. So there's going to be 3 differences that I highlight. This is one of them. So in cardiac muscle, the depolarization is prolonged because of the delay in opening the potassium gates and this calcium influx. Then after this plateau period, then the potassium gates do open and positive charges leave, and then we go back to our resting membrane potential. So that prolonged depolarization, that plateau, is one difference between cardiac and skeletal muscle. A second difference is that because the depolarization is prolonged, the contraction is prolonged. And so here we have a resting cardiac muscle cell, but we've depolarized, and now it will begin to contract. It will hold it and hold it and hold it and hold it for a quarter of a second. That's 250 times longer than fast twitch skeletal muscle. Because this depolarization is prolonged, the contraction is prolonged. So when cardiac muscle contracts, it's not a fast twitch where it contracts and relaxes. It contracts and holds it. And that's useful because we have this big left ventricle. And we don't want to twitch here and a twitch there, a twitch there, a twitch there. That's not going to pump blood to the brain and back. In the same way that in a tug of war, if everyone grabs the rope and this one gives a tug and then that one gives a tug and that one gives a tug, if they're not synchronized, at no point do you have enough force then to win the tug of war. And so what we want is if every cell is holding as their contraction for a quarter of a second, then we'll get them all acting together, which generates enough pressure to get blood to the brain and back. So the second thing that makes cardiac muscle significantly different in this sense from skeletal muscle is that because the depolarization is prolonged, the contraction is prolonged. The third thing is I'll typically ask a class, tetanus, is that good or bad? And most of the class will say bad. And then I remind them, you're not wrong, but you're also not entirely right, because in AMP1, one of the ways that the word tetanus is used is that instead of a muscle twitch, where a muscle contracts and relaxes, if you wanted to lift something heavy, you would then just grab it and hold. And then instead of contract, relax, contract, relax, contract, relax, the cells would say contract and just hold it and hold it and hold it and just maintain this, contraction. This is what we call tetanus. And in cardiac muscle, in skeletal muscle, that's a normal part of exercise. The muscles can undergo tetanus. In cardiac muscle, one doesn't want tetanus, because if I'm exercising, if I'm running and my left ventricle squeezes and squeezes and squeezes and squeezes, after a second, that's useless, right? Because once the blood has been squeezed out of the ventricles, squeezing more doesn't get any more oxygenated blood to my brain, my muscles, etc. So what I want to do is to relax so that it can fill, and then squeeze again, and then relax so it can fill, and then squeeze again. And so tetanus is a normal part of skeletal muscle, but tetanus does not occur in cardiac muscle. So before we get to the next part, just a quick comment. For AMP2 students, you spend hours studying sliding filament theories and action potentials in AMP1, and then you might wonder, like, do I have to do that all over again? And the answer is no. Most of that applies to cardiac muscle just as it does to skeletal muscle, but there are some differences. So for example, in cardiac muscle, because of the calcium influx and the delay in the potassium gates, the depolarization is prolonged. Because the depolarization is prolonged in this plateau, then the contraction is prolonged a quarter of a second as opposed to 1,000th of a second. And then thirdly, tetanus is not possible. So the next video will continue with the anatomy of the heart, going through the valves, the vessels, and the chambers.